Hey, Bio30s. Today we're going to be discussing the second form of cell division, which is meiosis. Now, meiosis is different from mitosis in a few distinctive ways. As you remember in our last video on mitosis, mitosis, its goal is to basically take a parent cell and to reproduce it into two identical daughter cells. In the case of meiosis, there are two things that meiosis is trying to do. One, it's going to try to create a situation where you've got genetic recombination going on. So the daughter cells are not going to be identical to one another, and they're not going to be identical to the parent cell. So that's one of the major goals of meiosis. And the other major goal is genetic reduction. Basically, you are going to take a cell that is, in the case of humans, a diploid cell, and you are going to convert that cell into a haploid cell. So for example, in a human, if we're producing an egg or a sperm cell, we're going to go from a parent cell that has 46 chromosomes down to an egg or a sperm cell that has only 23. Now, meiosis works a little differently from mitosis in another way, in the fact that there are two rounds of division. In mitosis, there was only the one round of PMAT division: prophase, metaphase, anaphase, and telophase. In the case of meiosis now, you're dealing with PMAT I and PMAT II, and I'll be discussing both of those processes today. All right, here we go. So, when we're dealing with meiosis in a human, it's pretty much only to create a gamete. That's the only time that meiotic division is going on in humans, is to produce either a sperm or an egg cell. And there is a somatic cell, which is known as a germ cell, and that germ cell would initially be a diploid cell, and it's going to begin two rounds of division in order to produce the gametes, which are going to eventually be haploid. Because there are two rounds of division, there will not be two daughter cells. There are going to be a total of four daughter cells being produced at the end of the two rounds of division in meiosis. So let's take a look at these. And these are going to look very similar to what we discussed the other day with mitosis, but you'll be noticing a few differences, which I'll try to point out along the way. So we're starting off again with a prophase division. So again, the cell has duplicated its DNA. We're basically with a cell, just like last time. This cell is a 2n equals 4 cell. So we have got two pairs of homologous chromosomes that have been duplicated into 8 strands of chromatin, which will then eventually become these 8 chromatids that are going to be connected together. So again, just like in prophase and mitosis, here's what's going to happen. You've got a nuclear membrane that begins to disintegrate. The centrals will begin to form with the spindle apparatus. The genetic material will begin to condense. Now, one thing I do want to mention, last time when I did the video on mitosis, all the genetic information, or the DNA, was coded blue. In this case, I've got blue and green. There will be a very good reason as to why I've done that in a couple of minutes here. I will explain why, but the genetic material is basically blue and green, and the blue-green would be a pair of homologous chromosomes that have been duplicated, which I'll get to in a second. So the chromatin, we've got 8 pieces of chromatin, which are going to condense into 8 sister chromatids. Each of those pairs of sister chromatids are going to be attached with a centromere. So each of those X's represents essentially a duplicated chromosome, which has been duplicated into 2 sister chromatids. So in prophase, you're going to see early prophase, nuclear membrane. By the end of prophase, late prophase, very little of the nuclear membrane is going to be left over, and you're going to see the genetic material going from chromatin to essentially duplicated chromosomes, or chromatids, sister chromatids. And the spindle apparatus and the centrals will now be on opposite sides of the cell for that first round of division. All right, so that's prophase 1. Metaphase 1 is going to look different than what we saw metaphase look like in mitosis. In metaphase, in mitosis, if you go look in your notes, you'll probably notice that the configuration looks different than what you're seeing in the diagram here. when I had a cell that was 2, 4 equals N during mitosis, each of these X's, each of these sister chromatid pairs were aligned in a row. So I had 4 of these X's sort of lined up along the center of the cell. In meiosis, the duplicated homologous pairs of chromosomes, that's a mouthful, otherwise known as a tetrad. So what we've got here is these. So let's say, for example, this would be chromosome pair number 1 in our karyotype, like I showed you guys in our video about genetic material at the beginning of the unit. Imagine this is chromosome pair number 1, and it's been duplicated. This would be the chromosome that the cell got from the mother. This would be the chromosome that the cell got from the father when the initial organism was made. They're going to line up together. So basically, each pair of chromosomes that have now been duplicated are going to arrange themselves into these formations that are known as tetrads. So the tetrad represents a pair of chromosomes, chromosome pair number 1 or chromosome pair number 2 in this case, that have been duplicated. So I've got 2 duplicated chromosomes, a homologous pair, that has now been duplicated into 4 chromatids. So there are 4 chromatids that represent the original 2 chromosomes in the cell. So originally we had 2n equals 4. We've got now 8 chromatids, but you see they're aligning in their pairs. Because what's going to happen next is very different than what happened in mitosis. So in mitosis, when we got to anaphase, the chromosomes or the chromatids actually separated. That is not the case in meiosis. You'll notice that during anaphase, the chromatids, the pairs of chromosomes, the original chromosomes that represent pair 1 and pair 2, they're being separated right now, but the chromatids are not being separated. You're also going to notice something else about the diagram, and I'm going to get to this more in the next video, which will be part 2 of meiosis, it'll be the next video you watch. You'll notice that they are no longer purely green or purely blue. We mentioned at the beginning of this video that one of the goals of meiosis is to create genetic recombination. What you're seeing here is what is called crossing over, and I'll discuss that more in the next video. But just for now, be aware of this, that during metaphase 1, the chromatids, the non-sister chromatids, are able to actually overlap one another and exchange genetic information. So these chromosomes that are eventually going to separate at the end of the 2 rounds of meiosis are not going to look the same as the original chromosomes that were in the original germ cell. They are going to be a combination. Like this cell here, this chromosome here is going to have bits of the original parental chromosome, or father chromosome, and part of the mother's chromosome will be connected into this new configuration of genetic material. And that's how meiosis does it, is through this act of crossing over, which I will explain more in detail in the next video. But when you're looking at images between, if you have to separate an image between mitosis and meiosis, one of the things you always want to look for, there's always going to be some kind of indication that there's been a crossing over event. There's going to be some kind of a colorational change. If the genetic material looks identical at the beginning of the process and doesn't look any different at the end, you probably have mitosis. But in this case, because the coloration has been changed, we have some green mixed in with blue, that's indicating that there's been a crossover event. There has been a genetic recombination of DNA happening during metaphase 1. So in anaphase 1, sort of the same idea as what happened in anaphase. In this case now, the chromatids, the tetrads are separating, but the chromatids are still connected. So we still cannot call these chromosomes yet. We can't. And then at the end of anaphase, when we get into telophase, you're going to see the pinching off of the cell. Nuclear membrane may or may not reform, depends on the type of cell involved, but we're going to go with the version where the nuclear membrane does reform. The centrioles basically disappear, the spindle apparatus disappear, and we get basically now, we're dealing now with, this cell right now is no longer diploid. This is now a haploid cell. And you might be saying to yourself, well, Mr. B, you mentioned earlier that this was a 2n equals 4 cell. And I did, and that's what it was. But you're going, well, Mr. B, there are 4 chromosomes here. No, there are not. Remember, these X's represent chromatids. And the 2 chromatids only represent a single chromosome that has been duplicated, right? So we still have to do what? What do you think we're going to be doing in the second round of division? We need to separate the chromatids. So after the second round of division, what I should wind up with in each of the daughter cells should be 2 chromosomes when I'm done, which would then equal that N equals 2. We're not quite there yet because the chromosomes, the chromatids, see, even I make that mistake. The chromatids have not separated yet. So we've got a haploid cell that is in a duplicated configuration. This would be like a cell entering into, the beginning of, mitosis. You've got duplicated chromosomes that have to be separated. That's what we're going to be doing in the second round of division. The second round of division is all about separating these sister chromatids from one another. Now, if you look at the second round of division, the second round of meiotic division, PMAT II, looks pretty much identical to mitosis. The only giveaway is this, is that you are going to see mixing of colors in the chromosomes. If you see chromosomes that look like there's swapping of colors amongst the chromatids of the chromosomes, you know you're not in mitosis country. You know you've got genetic recombination going on. That means you're in the second round of meiotic division, which would be meiosis II, which is PMAT II. So prophase, metaphase, anaphase, telophase II, which is going to lead eventually to our 4 daughter cells here at the bottom. So let's go through it really quickly one more time. What we've got right now is we've got our prophase II cells that are basically going to see our nuclear membranes fall apart. The centrioles will reform, spindle apparatus will reform, and the centrioles will move to opposite sides of the cell. During metaphase II, it looks identical to metaphase and mitosis. Your sister chromatid X's are going to basically line up all in a nice little row in the middle of the cell. In this case, we've only got 2 pairs of sister chromatids that are left, or I guess 2 X's left in each cell. And then when we get to anaphase II, this is the big moment when the chromatids finally separate. So in mitosis, the chromatids actually separate during anaphase, but in meiosis, the chromatids do not separate until we get to anaphase II. Remember, the tetrads separate during anaphase I. The chromatids actually separate during anaphase II. And when we're done that process, which looks very similar, like I said, to mitosis, what we're going to be left with now in our cells is, take a look, let's do some counting here. We've got 2 pieces of chromatin in this cell, 2 pieces of chromatin in this cell, 2 pieces of chromatin here, 2 pieces of chromatin here. If I bring back the original image of the original cell, and I'm going to do that right here. You'll see here that in that original cell, we had 4 pieces of DNA that had been duplicated. So we had 8 pieces of chromatin. So technically, the original cell before S phase and interphase, before that DNA was duplicated, had 4 pieces of chromatin. So that original cell was 2n equals 4. By the end of metaphase II, by the end of meiosis II, you've got a cell now that has only 2 pieces of chromatin. So these cells now are the 4 haploid cells that are the result of the meiotic division. You will also notice that each of the cells has a different configuration of DNA. You've got different recombinations of the genetic material. So none of these cells look like the parent cell, and none of them look like one another. So there's your ability for the genetic recombination, but you've also got your genetic reduction going on here because you've gone from a cell that was originally a diploid cell to now a cell that is haploid. If you want to take a look at a quick little video that might actually clear some things up for you, go click on that right now. Otherwise, we're going to keep on going here, and I'm just going to quickly show you a couple of diagrams here in the notes. Again, if you're still having some trouble, Hopefully you've got the note package in front of you, but I've got some great diagrams here of these stages. This is a meiosis 1, and then right over here you've got meiosis 2 going on, showing you the whole process that I just basically outlined on the board for you. Let's do a quick review. If you've got your notes in front of you, let's see if you can actually play along at home and see how this goes. If you've got mitosis, what cells are involved in mitosis? Those are going to be your body cells. Somatic cells are the ones that are going to be involved in mitosis. How about meiosis? What cells are created using meiosis? You should have said that it was going to be germ cells that were going to become gametes. So these are the only cells that undergo meiosis in the human body, are these germ cells, either in the testes or in the ovaries, depending if you're a guy or a gal. How many rounds of division are in mitosis? You should know this. Mitosis has 1 round of division, and meiosis has 2 rounds of division. How many the number of duplications of chromosomes. How many times are the chromosomes duplicated in mitosis? Well, there's going to be 1 duplication of chromosomes. That's going to occur. And remember, DNA does not get duplicated in M phase. A lot of people make that mistake, whether it's mitosis or meiosis. Remember, DNA does not get duplicated in M phase. It gets duplicated in S phase, and the same is true for meiosis. There's only 1 section or 1 time when the chromosomes become duplicated, and that is during S phase, before the meiotic divisions begin. What about the number of cells resulting from a complete divisional event? If it's mitosis, I should have how many cells when I'm finished? I should have 2. If it's meiotic division, I should have 4 daughter cells being completed at the end of my rounds of division. What's the chromosome number of the parent cell in a human? So if I have a parent cell that's undergoing mitosis, what would the chromosome number be of that cell? The cell's going to be a 2n equals 46 cell. What about in meiosis? It's also going to be a 2n equals 46 cell. Those germ cells, remember, are diploid. They are not haploid until you undergo meiosis. That's when they become haploid. What's the chromosome number in the daughter cells for mitosis? You should have said that it's going to be identical to the parent, so it's going to be a diploid 2n equals 46 cell after the rounds of cell division in mitosis. In meiosis, I'm going to have haploid cells that are going to be n equals 23. So my gametes and my gametes and my sperm and my egg cells, are going to have 23 chromosomes in them, respectively, at the end of meiosis. What kind of daughter cells am I going to get after mitosis? Daughter cells are going to be identical to the parent. They're going to have the same amount of DNA as the parent, and they're going to be identical in every respect. What about after meiosis? There are going to be daughter cells that have half the amount of DNA as the parent, and they are going to be genetically different from the parent, which is the whole point behind meiotic division. What's the function of mitosis? Several functions, actually. There's growth, there's repair, there's maintenance, there's also, it's used for asexual reproduction. Meiosis really only has 1 function, and it's used to create gametes for sexual reproduction. There really is no other use for mitosis in humans or any other organisms that produce gametes. They're only using meiosis to produce gametes that are going to be used for sexual reproduction. And again, you've got one last diagram here to take a look at. So great diagram here that is going to basically compare mitosis and meiosis, what they look like, how are they similar, how are they different, and that basically that covers our process of meiosis. And this ends the first of 2 videos about meiosis. Next time when I talk to you guys, it's going to be about how do we actually create the genetic recombination in meiosis. We're going to be discussing independent assortment, and we're going to be discussing crossing over next time. So until then, guys, take care, and we'll see you soon.